So we've looked at innate immunity, the first and second line of defense, and now we're going to look at adaptive defensive. So innate were the things that we're kind of born with. Adaptive are those things that we pick up or acquire along the way. So let's kind of take a review and look at where we're at. So if we start out with the pathogen or the microbe trying to gain entrance to the body, the first thing he's going to encounter is that first line of defense, those mechanical and chemical barriers, the skin, mucous membranes, flow of fluids, right? The ciliate or escalator, sweeping that out. So the first thing is our fence. We're trying to keep our fence up. So if it is able to get through that first line of defense and break through that first barrier, then it's going to we have two things we can do. We have our second line of defense, which we looked at, and that's really kind of more where the chemical barriers fall. Enzymes, pH, also remember our interferons and defensin, those proteins, collectins, our complement cascade. Then we had three things to turn it on and three consequences: inflammation, opsonization, and cytolysis, right? And then the three ways to turn it on were our classical pathway, our alternative pathway, and our manoselectin pathway, right? And so we looked at the natural killer cells, which were kind of the in-between guys that could kind of fall really in adaptive or innate. And then we looked at the three consequences of complement cascade, and that's inflammation, phagocytosis. Actually, that's just some different consequences here: inflammation, phagocytosis, and running fevers. We talked about all of those, okay? And then so now we're going to look at our third line of defense, and we're going to break this down into two areas, okay? We're going to have our cellular immune responses and our humoral immune responses, all right? So this is a really good overview. This is a good base diagram for you to start with. In class, I do a big color-coded diagram that's got pictures and about 60 different points on there, which is usually a big essay, all right? And so this would be a good place to start. List these out and organize them, and you actually going through and organizing this material yourself instead of me just giving it to you and you spit it back is actually going to help you to learn this much better. All right, so figure out, where do those white blood cells belong? Where do those antibodies belong? Those types of things. So our adaptive defenses, remember, these are our specific defenses. We have specific antibodies that target specific antigens, and this is what we typically think of when we talk about immunity. It's our third line of defense, all right? Remember, adaptive is split into humoral and cell-mediated, okay? So immunity, this is a resistance to a particular pathogen or a toxin or a metabolic byproduct that sometimes a pathogen releases, all right? So a gram-negative bacteria, for instance, when it dies, its body actually can make us very sick even after we kill it. Okay, and so that would be kind of the toxin side there, all right? And so this is based all on our ability of our body, the ability of our body to be able to recognize self and non-self. And it does this on the basis of what we have. It's kind of like little name tags, little proteins, we call those antigens, that are on the outside of a cell. And they will have a particular shape. Okay, and remember, shape determines function. So shape of the antigen is how we recognize this as self or non-self. Now, sometimes our body doesn't work correctly, okay? And so what can happen is we can have self cells that somehow, either sometimes due to an association with a particular virus, our self cells get tagged as non-self. And so then our body actually begins to attack itself, all right? And what is that? That is an autoimmune disease, okay? So in an autoimmune disease, the immune response is directed towards the cells that belong in the body, and the body has just forgotten what they look like. An example of this is systemic lupus erythematosus, and you probably just are familiar with this as lupus. It's an autoimmune disease, and it affects skeletal muscles, and they get tagged the wrong way, basically. So let's talk a little bit more about antigens. Do you remember what an antigen is? We said an antigen was basically any foreign substance. That can be a microbe like salmonella, typhi, methicillin, I'm sorry, Staph aureus. I was trying to get from MRSA to Staph aureus. It can be something like pollen, okay? And so antigens can be those proteins that I was talking about, and we can sometimes call those epitopes. They can also be polysaccharides, so large sugars, glycoproteins, so a sugar-protein combination, or a glycolipid, which is a sugar-fat combination. And the most effective antigens are going to be large and complex, all right? We also have what we call haptens, and these are small molecules that are not necessarily antigens by themselves, but if they combine with a larger molecule, then they can sometimes still stimulate or trigger an immune response. All right, now we're going to look at our T cells and the cellular immune response. Remember we said cellular and humoral, so we're looking at cellular right now. So a lymphocyte must be activated before it can actually respond to an antigen. So we kind of have to put them into action. We got to send the lymphocyte to do its job. So T cell activation requires what we call an antigen presenting cell. And this is kind of, it says accessory cell. This is a helper cell, and we even call them helper T cells, okay? They include things like macrophages, B cells, and then there are some other types of cells as well. All right, and so they basically help to trigger the response and to activate and to let the T cells know that they need to come into action. This requires what we call a major histocompatibility complex, okay, or human leukocyte antigens to be recognized as non-self, which is basically like a name tag. All of our cells have name tags on them, and they're going to be these MHCs or these HLAs, and we're going to recognize them as self or non-self, okay? T cells then can make and release polypeptides that we call cytokines. And you may have heard of cytokines before, where you can actually have a cytokine storm, and that's where the inflammation kind of gets out of control and becomes a bad thing instead of a helpful thing, all right? But we have lots and lots of different types of T cells. I can't remember the exact number, but I want to say there's like 200 different types. Don't quote me on that. I might be wrong on that one. But the main ones we want to look at here are the helper T cells, the cytotoxic T cells, and memory T cells, okay? In the picture here was some T regulatory cell, but we're going to focus on memory T cells. So cellular immunity was mediated primarily by T lymphocytes inside infected cells. Humoral immunity is more serum proteins, and we call it antibody mediated. And the B cells are actually producing antibodies. So B cells are activated when an antigen fits with the shape of its receptor. And so again, we've got shape and structure determining function. They must fit together. You can kind of see this here in that diagram right there. One of the really cool things about our immune system is we already have pretty much any antibody shape that will fit any antigen that we could come into contact with, which is pretty phenomenal to me because we have so many of them there just sitting and waiting. The thing is, we have to increase them and make them into memory cells to be immune, but we have one there ready and waiting to start that process that will be able to handle pretty much anything that comes against us, which is pretty amazing to me. So B cells are activated when that antigen fits the shape of its receptor. It does sometimes require those T helper cells to activate them and call them in. They're going to release cytokines, okay? And those are going to stimulate the B cells into action. Sometimes those B cells then go on to become the memory B cells because they're going to differentiate into plasma cells that are going to produce and secrete antibodies, okay? So in the cellular immune response, T cells attach directly to the antigens and destroy them. Antibodies are produced. We call those immunoglobulins, all right? Immunoglobulins. Humoral immunity's main goal, its main function, is to create those antibodies, those immunoglobulins, right? So our most valuable guy here, our MVP, is the B lymphocyte because he is the one that makes antibodies. So what do antibodies do for you? Fight off infection? Okay, so how? They don't directly fight infection most of the time. That is one method, but not directly. But usually what they do then is they make a wanted poster in our body. And that allows us to recognize and tag them as antigens. Yep, that's opsonization, okay? We tag them as a foreign contaminant. And so that somebody else can go in and do the dirty work, right? So opsonization is a main goal here. We're going to tag it as foreign so it can actually be attacked by other cells like killer cells, natural killer cells, okay? So antibodies then can actually turn on complement. They can trigger complement. Remember, we had that one exception that we said was antibody, included antibodies, all right? And one effective complement then is cytolysis, so they can actually kill through complement. Antibodies can surround the bad guy, and if you're surrounded, you can't bind with any, right? So you can be rendered where you can't do anything. I can't think of the right word at the moment. Rendered useless, basically. All right, so if we are surrounded, you're not dead, but you can't really commit any crimes either, can you? Okay, and when we surround them, then that is agglutination. Okay, agglutination, cells clump and stick together. So we have surrounded them and they are ineffective. All right, we hadn't got rid of them, but they can't do anything. And so then we have our natural killer cells, right? Remember, these are the only non-specific lymphocytes. We have B lymphocytes that can sometimes make antibodies themselves. But remember, sometimes they need a helper, and that's the T helper lymphocyte. It comes in and turns on and says, hey, go ahead and make some antibodies. They're not required all the time, but they do assist with production of certain immunoglobulins, okay? Cellular immunity, in a large part, relies on cytotoxicity. This is the backbone of our immunity, our T lymphocytes. They are our backbone, okay? And more specifically, our cytotoxic T lymphocytes, okay? So they're really important. These are kind of like the surgeons of your lymphocytes. They want to come in, show up in the ER, have all their tools out, the patient is ready, they want their x-rays and blood work, patient's already under anesthesia, and they just want to go in, do the surgery, and get out and be done, okay? And so then they'll leave the prep work and the aftercare to somebody else. And so this is what our CD8 T lymphocytes are like. Okay, and this is a class of cytotoxic T lymphocytes. These are clusters of differentiation. That's what CD stands for, clusters of differentiation, and they are cytotoxic T lymphocytes. They require a lot of support, right? A surgeon comes in and you've got everyone from CNAs, from aides, from maintenance where they're cleaning up before and after and sterilizing, and then you have your surgical nurses in there, you have your anesthesiologists in there, so they have all kinds of helpers, okay? They require support. And those support guys are the helper T lymphocytes. So CD4 are helper lymphocytes as well. So this is the cell that actually HIV targets. It targets the CD4 cell and actually takes advantage of that. And we'll talk about that a little bit later. It takes advantage of that to actually gain entry to the cell in our body and move around. We then, of course, have our antigen presenting cells, and these are the guys that go and show the wanted poster signs. They show wanted poster signs. They could maybe perhaps say, you could say, make the diagnostics for the cytotoxic T lymphocytes, and the guys that do that are the macrophages and dendrocytes. So that's important to remember here. Macrophages and the dendritic cells, those are the ones that are the antigen presenting cells typically. Let's look at the different types of antibodies. We've got 5 different types of antibodies, and this is medically important because you can test for one of two of these antibodies to see if somebody has had something like COVID-19, right? Are you making antibodies to this? Antibodies are also what we call immunoglobulins, all right? So the first type of these is IgG. IgG, immunoglobulin G. Say that 5 times really fast. This is the main antibody found in blood serum. This is a longer acting one. It takes about 14 days, and you usually see this with chronic conditions, okay? It's the most abundant type, and it can have an anamnesia memory response. Anamnesia. Amnesia is where you forget something. Anamnesia is where you remember something. So it's a memory response. So that's protective in the future. IgM antibodies are also found in blood serum. These are more short-term. They last about 5 to 7 days, and these are like the first responders. So these are the first to show up on the scene, okay? The first guys to show up on the scene. Next is IgA, and these guys are found in mucous membranes. There's a little bit of it in the serum. These are shorter acting as well, around 5 to 7 days. They are not quite as specific as the other antibodies, so they can kind of bind to things that aren't necessarily a perfect fit. They can ward off viruses that are similar but slightly different than the ones that they fit perfectly with, like if you were to get, for example, a flu vaccine. IgE is a hypersensitivity working antibody. This, you see lots of this elevated when you have things like allergies. This will stimulate inflammation. This is a component of anaphylactic shock. And then lastly, we have IgD, and these are found on B lymphocytes, and this helps remember the pathogen and be a memory and react much quicker the next time it comes into contact. B lymphocytes can also remember, sometimes make antibodies themselves, but sometimes they need those T helper lymphocytes to come in and turn on that antibody production process. So the structure of antibodies are they have a heavy region and a light region, and they have a variable or different number of binding sites. They have from 2 to 10 binding sites. You can see up here, these are the binding sites. So here we have 2, okay? And so we've got 2 colors here that are more heavy chain, so that's these guys. And then we have 2 colors here that are more of a light chain. So how do we classify the different types of immunity? We have 2 basic ways. We have natural and we have artificial. So did I actively make it or did I passively make it would be the other way. So natural or artificial, and then active or passive. And then we kind of put those together in groups. So what we want to look at is the route of exposure. Did I make these myself? Or did someone give them to me? Like humans can pass antibodies across the placenta to the offspring, or in the colostrum. So newborns can acquire passive immunity through things like breast milk. Injections of gamma globulin are sometimes given to provide artificially acquired passive immunity, which gives a boost to patients for the short term, like if you're getting a kidney dotation or if you have hepatitis A. All right, let's classify what we call a hypersensitivity. And we have 4 types of hypersensitivities. And these are basically when your body responds overzealously to a substance. So type 1 hypersensitivity, this is where your body reacts. And this uses the antibody. This is affected by IgE. IgE. So This is anaphylaxis, all right? And so when you hear anaphylaxis, you're probably thinking of anaphylactic shock, which is life-threatening. But anaphylaxis is a overgeared reaction against poisons in foods and allergens. You can actually have a focal anaphylaxis, like something against pollen, hay fever, seasonal allergies. This is a typical type 1 sensitivity. This is going to be IgE antibodies, food allergies, wasp stings, peanuts, all right? This occurs in minutes after contact with the allergen. Can cause things like asthma attacks, eczema, and yes, anaphylactic shock, redness and swelling to kind of an extreme degree when you get stung by, say, a bee or a wasp. Next, we have type 2 hypersensitivities. These are antibody-dependent cytotoxic reactions. These guys are going to be IgG or IgM. And these target something that is recognized as foreign. So this could be a blood transfusion reaction, okay? So if you've got positive type blood and negative type blood and you give one the other, they can have this blood transfusion reaction, okay? So IgG and IgM play a role in these. Type 3 hypersensitivities involve IgM, let's find a color here, IgM, or that's a G, IgD, all right, IgM or IgD. So this is an immune complex reaction. Basically, antibodies are going to clump together and cause this big conglomerate and clot. And this is the problem. So you get big chunks of antibodies of IgM and IgD. And this is what an autoimmune disease does. Your body no longer recognizes some parts of itself and attacks it. So like, for instance, in lupus, which we talked about, you get arthritic degenerative changes because you have big chunks of antibodies, clusters of antibodies that sit in your joints. And it's kind of like having pea gravel in your shoe. You go out for a run, not good feeling, right? You got to get that out of your shoe. And so it rubs in between those joints and starts to tear things apart, okay? It degrades structures down. So it damages those body tissues. Type 4 hypersensitivities are a little bit different. Instead of antibodies, these are T-cell dependent. T-cell dependent, okay? So these are delayed allergic reactions. These T cells come all the way from the thymus, and so it takes a while for them to get there. They have to travel, so it's delayed. So after a long time, you've been wearing latex gloves that have powder in them, and you've been wearing them for months, and then all of a sudden they start to bother you. That's a delayed reaction allergy, okay? We use this in the test for tuberculosis, all right, Mycobacterium tuberculosis. This is one of the #1 infectious diseases in the world. And so you're exposed to a bacteria for a long period of time, and then you develop a T-cell response to it. So to test you for it, they're going to put a small amount of tuberculin in your skin. If you have a response and swell up large enough, you'll have a positive TB test. So that would be a type 4 hypersensitivity. It takes a while to develop. Another would be transplant reactions. Why is it some people can live with a transplant for months and then all of a sudden reject their transplant? It is a delayed reaction hypersensitivity type 4 reaction. These take at least 48 hours to occur, but as you know, with transplant, it can be months later on down the road. So transplant recipients, the body rejects that transplanted organ because of a mismatch in tissue type. That mismatch in tissue type is based on that ID tag we talked about that is on the outside of cells, that major histocompatibility complex. That's MHC, major histocompatibility, all right, complex. So this is sometimes called human leukocyte antigens, and these help helper T cells, they help T cells recognize a newly displayed antigen as a foreign substance. Tissue rejection looks like a cellular immune response to a foreign antigen. The more alike the antigen surface molecules are, the more likely it's going to be accepted. The greater the difference, of course, the quicker and the faster rejection can occur. And we use immunosuppressive drugs in order to prevent that rejection. So we have to suppress this portion of the immune system to keep it from fighting against the newly transplanted tissue or organ. So we've got four main types of grafts or tissue transplants. If I have, say, a graft from my identical twin, then that would be an isograft. Iso means the same. So would I reject something that's the same as me? No, I wouldn't. The DNA is the same, right? If I take a graft from myself, auto means self, so would I reject that? No. Auto is self, so that's if you take skin, for instance, from your back and put it on your arm or a leg after a burn, okay? Allo is where you are taking an allograft. Allo is the same species. So this is where you would get a graft from not yourself, not your identical twin, but another human being, all right? And so this could be something you got from a cadaver. You could have received something from a cadaver, perhaps. And then we also have, and this is a new and really a very interesting area, xenograft. Xeno is going to talk about a broader scope of things. You talk about xenophobia. Well, that's about being afraid of people from other places. So xeno means other. So a xenograft is from another species, like say getting valves from a pig heart, that sort of thing. And there are some really fascinating things that we do now in order to have this graft occur for xenografts. So we've already kind of touched on this, but this is a really good chart in your textbook 16.1 of some of these different autoimmune disorders that you want to be familiar with. Remember, an autoimmune disorder, auto meaning self-immunity, we no longer recognize cells that are supposed to be there as self. So we recognize them as foreign invaders. We don't recognize our own family anymore. And then so the body produces antibodies, and we call these autoantibodies. And then the cytotoxic T cells actually go and attack your body's tissues and organs. And these can be something as minor as like eczema or as bad as like lupus, multiple sclerosis, myasthenia gravis, okay? And I believe there's a few of these on your clinical list of videos to watch. So be sure and watch those and look over this list because it's important to understand which of these are autoimmune disorders. HIV, this is a big clinical application here, and I am fortunate enough to be probably older than most of the people watching this video, most of my students. But the only reason I say I'm fortunate enough, and actually this is not a fortunate, it's actually kind of a sad thing, it's, well, terrible, actually, horrible, but I do remember when AIDS was first discovered, okay? I remember when this first came to America and how scared everyone was. And they actually called it gay cancer because when it came to America, it targeted a population of people and was spread throughout that population of people due to lifestyle choices, okay? And so it was targeting young homosexual men, and so they were just puzzled, and so for a long time they called it gay cancer. And what helped them to actually figure out what was going on, because basically part of the immune system was just completely obliterated and wiped out. And so all these opportunistic infections would start coming into play in young, healthy individuals that normally did not get these infections. And that is actually what helped them to kind of identify what this was and figure out that it is actually HIV, okay? HIV is the human immunodeficiency virus. So it's a virus that attacks the immune cells, and then it goes on later to cause what we call AIDS, and that's acquired immune deficiency syndrome. So that's where you are no longer able to fight anything off. all right? HIV is a virus that has reverse transcriptase, and it actually is what we call a retrovirus. It makes DNA from viral RNA. All right, viruses are non-living. They enter the body, take over the body's own machinery in order to replicate themselves. They cannot copy themselves. They cannot replicate. And so the first thing that has to happen is it has to cross that first line of defense, the mucosal barrier, right? And so then it gets into a macrophage. And then later it targets the T helper cells, right? And what it does is it takes advantage of this protein called GP120 on that CD4 helper cell in order to gain entry into the cells. And remember, those CD4 cells go around to other cells, all right? And so they are taking it around to other cells. And so it has gained access that way. It breaches our barriers and it affects immunity. It enters the macrophages. Then it enters the helper T cells. And then the helper T cells go to the cytotoxic T cells to try to show them that we have a foreign invader, and then they get into the cytotoxic T cells. And so the helper T cells begin to die off first, and bacterial infections begin to start rapidly, okay? HIV has a very, very high mutation rate. Last I heard, it was about 50%. And so it is very, very difficult to treat because it is constantly evolving and changing. And so we have come up with this complex cocktail of antivirals and medications that they give now, which has really increased the long-term life, the ability to live for a much longer time than when it was first discovered. All right, so we still don't have a cure for it though, and this is a horrible, horrible disease. It completely wipes out the immune system. So finishing up on the HIV thing, I don't want people to be misled in thinking that HIV is only in homosexuals. It's actually the opposite. It started here because that was kind of the population it entered, but in other countries and places like Africa, it is primarily heterosexuals. Heterosexual men have it and tend to give it to women. So don't misconstrue that. That is just the way that it kind of played out in America. And so throughout our life, our immune system does decline and it gets weaker, especially in the elderly. So we become more prone to be able to have a higher risk for things. It declines early in life. The thymus gland starts to shrink at least by 25%, okay? The antibody responses get slower. Things, as we get older, we slow down, and that's on a cellular level as well. IgA, IgG antibodies increase, but the IgM and IgE actually decrease. And so a lot of times elderly can't take medications that suppress immunity. That is not going to work for them because they're already more prone to susceptible and their immune system has changed. And so it also changes at the beginning of life, I should say. The acquired immunity is built up throughout the first years of life. And then so on each end of the spectrum, that's why we have more vulnerable populations.